Welcome back to the Deep Dive. Today, we're really diving into something huge in modern medicine. It's this whole molecular diagnostics revolution, driven by next-generation sequencing or NGS, and then, crucially, the bioinformatics needed to actually use all that data.
Yeah, it's essential. We cover this, the sheer number of techniques out there now.
Yeah.
It's pretty overwhelming for clinicians, for pathologists. I mean, molecular diagnostics isn't new. We had karyotyping, southern blotting back in the late 20th century. But now, it's just a dizzying array.
Right, and that complexity must make choosing the right test for a patient really challenging.
Yeah.
But you're saying NGS is the big shift here, the game changer.
Absolutely. It's the major technological leap that's reshaping the landscape. It's pushing out those older methods, the ones that looked at just one thing at a time, low throughput stuff.
And it happened pretty fast, didn't it?
Yeah. It really did. I mean, the groundwork was Sanger sequencing back in 77, then PCR in 83. Fundamental stuff. But this jump to massively parallel sequencing, NGS, that really kicked off in the late 90s and accelerated incredibly quickly.
The reasons are pretty clear, right? Speed and cost.
Exactly. Speed and cost. It's almost routine now to sequence a whole human genome in, a few days? And for under $1,000. That was science fiction not long ago.
Yeah. So this affordability, this speed, it means testing nucleic acids, DNA, and RNA, is now really central to managing, almost any disease.
Yeah.
Okay, let's unpack this. Before we get into the nitty-gritty of how NGS works, maybe we should clarify what we're actually looking for, these genetic markers.
Good point. Yeah, that distinction is fundamental, and the source material lays it out well. It really boils down to where the marker is found in the patient, because that dictates what kind of sample you need. Is it blood, tumor tissue, a simple swab?
Okay, so what are the main categories?
Two main types. First, you've got constitutional markers.
Constitutional, meaning?
Meaning they're present in essentially every single cell in that person's body. They were either inherited or maybe arose very early in development. Think cystic fibrosis, the CFTR gene mutation, that's constitutional.
Right.
So you can test for that reliably using, say, white blood cells from a blood draw.
Got it. And the second type?
That would be somatic markers.
Somatic.
These are acquired changes. They're restricted to specific tissues or maybe just a lesion like a tumor. Cancer is a classic example here. Mutations in genes like RAS, those are somatic.
Ah, okay. So they're in the tumor cells, but not in the person's normal, healthy cells.
Precisely. Which means for diagnostics, you absolutely need a sample of the tumor itself. You can't just use blood for that kind of test usually.
Okay, that makes sense. So for this deep dive, our goal really is to walk you, the listener, through how NGS generates this, frankly, staggering amount of data, and then how this field, bioinformatics, actually makes sense of it all, translates those raw sequences into something clinically useful.
Exactly. From data deluge to diagnostic insight.
So to really appreciate NGS, we probably need to talk about the limitations of what came before it, right? Like Sanger sequencing.
Definitely.
Yeah.
Sanger was the gold standard for decades, meticulous work. But it had a huge limitation. It needed a single, clean, homogeneous DNA template.
Homogeneous?
Yeah.
Meaning all the DNA molecules in the sample were basically the same in the region being sequenced.
Pretty much, yeah. So it gave you an average result for that DNA region.
And the problem with an average is?
Well, think about a tumor biopsy. It's almost never pure tumor cells, right? You've got normal cells mixed in, stromal cells. If the mutation you're looking for is only in, say, 10 or 15% of the cells in that biopsy.
The average gets skewed.
Exactly. The signal from that minority variant gets diluted, blended in with the normal sequence signal. Below about 20 or 30%, abundant Sanger just couldn't reliably see it. The result becomes messy, uninterpretable. It reads the bulk signal.
So for cancer diagnostics, where you often do have that mix, Sanger's unideal.
Often totally inadequate, yeah.
What about PCR? Polymerase chain reaction. That's still fundamental, isn't it?
Oh, absolutely. PCR is everywhere. It's often used as a step before NGS, actually, to enrich for the specific DNA regions we want to sequence. It's fantastic for making lots of copies of short DNA fragments, usually under 1,000 base pairs.
But PCR alone doesn't solve the heterogeneity problem for sequencing, right?
No, because standard PCR amplifies everything in the tube together, and then if you tried to sequence that bulk product with Sanger, you'd hit the same averaging problem.
Okay. So what makes NGS fundamentally different? Why is it better for these mixed heterogeneous samples?
The core difference is how it handles individual molecules. It gets around that signal blending issue. NGS isolates and reads individual DNA fragments, millions of them, all at the same time.
And that gives it much higher sensitivity.
Massively higher sensitivity. It allows us to reliably spot mutations even if they're present in only a tiny fraction of the input DNA, like 1%, maybe even lower sometimes.
Which is crucial in cancer, where a mutation might only be in a small subclone of the tumor, or the sample itself is mostly normal tissue.
Exactly. It's a clinical game changer for that reason alone.
So if PCR is still involved, maybe in the enrichment step, how does NGS itself avoid the pitfalls? You mentioned isolating molecules.
Right. NGS doesn't necessarily skip amplification altogether, but it changes how and where it happens. It's not about bulk amplification. Most NGS platforms rely on 3 sort of common principles, even if the specific chemistry varies a bit.
Okay, what's the first principle?
Spatial separation. This is the key to beating heterogeneity. You have to physically isolate the individual DNA molecules you want to sequence from each other.
How do they do that? Like physically?
A common way, for example on platforms using flow cells, involved attaching special adapter sequences to the ends of the fragmented DNA from your sample. Then these adapted fragments are flowed over a surface, maybe a glass slide or flow cell, that's coated with complementary sequences. The fragments stick, immobilizing them. Crucially, they stick sparsely enough that each original fragment occupies its own distinct spot. One molecule, one spot.
Okay, separated. What's next? Principle 2.
Local amplification. A single DNA molecule just doesn't give off enough signal, like fluorescence, to be detected reliably by the imaging systems.
So you need more copies.
Right, but you need to amplify it in place, right there, at that spot where the original molecule landed. This is often done using something called bridge amplification. It's basically a limited number of PCR cycles that happen right on the surface. It creates a small, dense cluster of identical DNA molecules, all originating from that single starting molecule, all attached at that same physical location.
So you boost the signal locally without mixing it up with signals from other spots.
Yeah.
You maintain that initial separation.
Exactly. You've kept the heterogeneity sorted out spatially, but now you have enough signal at each spot to read it.
Which leads to principle 3.
Parallel sequencing. This is the massively parallel part. Now you have millions of these distinct amplified clusters on your flow cell. The sequencing happens simultaneously for all of them.
How does the actual sequencing chemistry work in this parallel way?
Typically, it involves cycles of adding fluorescently tagged nucleotides, the A, T, C, and G building blocks. In each cycle, only the correct nucleotide can incorporate onto the growing DNA strand within each cluster. It glows a specific color. Then, a high-resolution camera takes a picture of the entire flow cell.
Capturing the color at every single cluster location.
Yes. So in one cycle, it figures out the next base for millions of fragments simultaneously. Then you wash away the fluorescence signal, add the next round of tagged nucleotides, and repeat. Cycle after cycle.
Hundreds of times.
Hundreds of times, yeah. Building up the sequence, read base by base for every cluster in parallel. This generates millions, sometimes billions, of independent sequence reads. They tend to be relatively short, though, usually less than 500 base pairs.
That mechanism makes the massively parallel concept much clearer. It's literally millions of tiny sequencing reactions happening side by side. But you mentioned the reads are short, under 500 bases. Does that cause problems down the line?
Oh, absolutely. That's a really important limitation to understand, and it's why, frankly, some older methods haven't completely disappeared.
How so?
Short reads are great for speed and accuracy on small variations, but think about the human genome. It's full of repetitive sequences, large, complex regions. Trying to figure out exactly where a short 500 base pair read fits, especially if it lands in a region with thousands of repeating A's and T's, for example.
The alignment becomes ambiguous. The computational tools struggle to place it uniquely.
Precisely. The algorithms can get confused. Is it from this repeat region or that identical one over there?
And this struggle, particularly with placing reads across large gaps or complex rearrangements. That's why some things are still tricky for standard NGS.
Yes. As the source material points out, certain kinds of genetic changes we're talking about, deletions or insertions spanning thousands of bases, or big gene rearrangements, or those tricky trinucleotide repeat expansions seen in diseases like Huntington's.
Right.
Those can still be very challenging for short-read NGS to detect reliably. You might need older methods like Southern blotting or specific hybridization assays like FH to really nail those down.
So it's not quite a one-size-fits-all replacement yet?
Not entirely. The sensitivity of NGS for single base changes or small insertions, deletions is phenomenal, unbeatable. But its ability to resolve large-scale structural variations is limited by that short read length. So yeah, the transition away from all the legacy techniques is happening, but it's gradual, and it depends heavily on what kind of genetic lesion you're hunting for. Test selection is still key.
Okay, so we've established NGS as this incredibly powerful engine, churning out sequence data at an unbelievable rate. High-throughput machines, they can easily generate, 400 billion base pairs of sequence or more in a single day.
That number is just hard to even comprehend. 400 billion letters of genetic code per day.
It is, and that sheer volume means the engine is useless without, well, a navigation system and a massive data processing center. That's bioinformatics. If NGS generates the data, bioinformatics is absolutely essential to turn it into something meaningful.
You can't just print out billions of letters and read them.
Not a chance.
Yeah.
Interpreting this flood of data requires specialized training, serious computational power, sophisticated algorithms, the whole 9 yards. So for someone trying to grasp this, how do we break down that computational process? How do you go from billions of short reads to, say, a clinical report?
We usually think about it as a pipeline, a series of computational steps. Broadly, there are 3 main stages.
Okay, stage 1.
Alignment. This is the first big hurdle. You've got all these millions, maybe billions of short, raw sequence reads. You have to figure out where each one belongs in the context of the entire human genome.
So you're mapping them onto a known reference genome, like a standard human DNA blueprint.
Exactly. You use these very complex algorithms to find the best fit for each short read against that reference sequence. The goal is context. You need to know which part of the 3 billion base pair genome each tiny read actually came from.
Makes sense. Once everything's mapped, what's stage 2?
Variant calling. Now that the reads are aligned, the software systematically compares the patient's sequence data at every position to that reference genome. It's looking for differences. Did the patient have a T where the reference has a C? Is there a small insertion or deletion?
And how does it decide if a difference is real or just a random sequencing error? Because errors must happen, right?
They do. The key factor here, the absolute linchpin for accuracy, is sequencing depth.
Depth, meaning how many individual reads cover that specific spot on the genome.
Precisely. Think about it. If only one read shows a difference, could be an error. But if 10, 50, 100 independent reads all consistently show the same difference at that exact spot,
then you can be much more confident it's a real variant in the patient's DNA.
Exactly. And the required depth depends heavily on what you're looking for. For a constitutional variant, something inherited, present in roughly 50% of the DNA molecules, a depth of, say, 30 to 50 reads covering that spot might be enough.
But for cancer, looking for those somatic mutations, that might be rare.
Right. If you're hunting for a mutation present in only 1% of the cells in that tumor biopsy, what we call a low variant allele frequency, or VAF, you need much, much higher depth.
Why? To be sure that one read in 100 isn't just noise.
You got it. You need statistical power to distinguish a true low-frequency variant from background error. Clinical cancer panels often aim for depths of 500x, 1000x, sometimes even higher at really critical gene locations, hotspots. So the variant calling algorithms weigh the evidence, the depth, the quality scores of the reads, and make a call only if there's enough statistical support to say, yes, this difference is real.
Okay, aligned, variants called. What's the final stage? Stage 3.
Variant annotation and interpretation. This is where we bridge the gap from raw data to clinical meaning. You might have a list of hundreds, even thousands of called variants. Now you need to figure out what they mean.
Adding context.
Exactly. The pipeline adds layers of information. What gene is affected by this variant? What's the predicted effect on the protein? Does it create a stop signal? Does it shift the reading frame? Then, crucially, it consults external databases. Massive databases that catalog known variants.
To see if this specific variant has been seen before and classified.
Right. Is it known to be benign, just normal human variation? Is it known to be pathogenic, clearly linked to disease? Or is it a VUS, a variant of unknown significance?
The dreaded VUS.
The bane of clinical reporting sometimes, yes. This annotation step combines computational lookups with human expertise, especially for interpreting those tricky VUSs or novel variants.
And this annotated list is what goes into the clinical report.
Essentially, yes. The final report that goes to the clinician highlights the variants deemed clinically significant. And often, especially because genomics moves so fast, the report will include a description of the biological impact, the clinical relevance, maybe even links to potential therapies or clinical trials based on those specific pathogenic variants. It helps guide the clinician who might not be a deep genomics expert.
So the bioinformatics pipeline is really doing the heavy lifting of sorting, comparing, and contextualizing.
Immense heavy lifting. But it doesn't necessarily stop at just listing individual variants. There are even more advanced applications.
Oh, way.
Well, think about it. If you have sequence data from the entire genome, or even just the exome, you have a huge data set. Can you see patterns beyond single base changes?
Patterns. What kind of pattern?
This is where things like mutational signature calling come in. It's a really fascinating area of bioinformatics.
Mutational signatures. How is that different from just finding variants?
Standard variant calling identifies what the change is. C to T at this position, deletion here. Mutational signature analysis looks at the types of mutations occurring across the genome and the specific sequence context around those mutations. It's looking for statistical patterns or signatures.
And these patterns can tell you something about the cause of the mutations.
Yeah.
Like molecular forensics.
Exactly like forensics. Certain processes that damage DNA leave characteristic scars, specific patterns of mutations. For instance, there's a well-known signature associated with damage from ultraviolet light exposure. Lots of C to T changes, specifically at sites where two pyrimidine bases are next to each other.
So you could potentially see evidence of past sun exposure in a skin cancer genome.
Precisely. But where it gets really clinically powerful today is in identifying signatures related to defects in the cell's own DNA repair machinery.
Ah, like if the cell isn't fixing its mistakes properly.
Yes. The classic and clinically vital example involves defects in the mismatch repair system. When mismatch repair fails, it leads to a specific pattern of mutations accumulating, particularly in short, repetitive DNA sequences called microsatellites. This results in a state called microsatellite instability, or MSI.
And detecting MSI is important because
Because it has direct therapeutic implications. We now know that cancers exhibiting MSI, meaning they have this underlying mismatch repair defect, are often highly responsive to a specific class of immunotherapy drugs called immune checkpoint inhibitors.
Wow. So the bioinformatics, by detecting that MSI signature from the NGS data, can directly point towards a potentially very effective treatment.
Absolutely. It's a prime example of how deep bioinformatics analysis extracts information far beyond just a list of mutations, directly guiding personalized therapy choices based on the fundamental biology of the tumor revealed by NGS. This is computationally intensive stuff.
Okay, so NGS plus bioinformatics gives us this incredible diagnostic power, and it's being used widely now, right, for both inherited diseases and cancer.
Very widely, yes. It's becoming standard of care in many areas. But for a clinical lab, there's always this balancing act. You have to choose the right NGS test for the specific clinical question.
Balancing what factors?
Things like, how much of the genome do you need to sequence? That affects cost. How deeply do you need to sequence? That affects sensitivity, especially for low-frequency variants. How fast do you need the result? And how much bioinformatics effort will interpretation take?
Right, it's not always about doing the biggest possible test.
Definitely not. Which is why the most common approach you'll see in routine clinical labs today is targeted sequencing, often using gene panels.
Targeted.
Yeah.
Meaning you're not sequencing the whole genome, just specific bits.
Exactly. You pre-select a set of genes known to be relevant to a particular condition. Maybe it's a panel of 50 or 100 genes associated with inherited cardiomyopathies or congenital deafness. Or, in cancer, maybe a panel of several hundred genes known to harbor actionable mutations or have prognostic significance.
And how do you target just those genes? How do you fish them out?
There are two main methods used to enrich for these sequences of interest before you put them on the NGS machine. It's important to know the difference. First, there's hybrid capture.
Hybrid capture.
Yeah, you design custom DNA or RNA probes, think of them as bait, that are complementary to the gene regions you care about. You mix these probes with the patient's total fragmented DNA. The probes hybridize, or stick, only to your target sequences. Then you use chemistry, often involving magnetic beads attached to the probes, to physically pull out just those targeted DNA fragments.
Like fishing them out of the genomic soup.
Exactly. Capture is very flexible. You can design probes for almost any region, and it works well for larger target areas, even up to the entire exome, as we'll discuss. It can be a bit slower, though.
Okay, and the second enrichment method?
That's multiplex PCR.
Multiplex, meaning many PCR reactions at once.
Yep. Instead of fishing, you're amplifying. You design hundreds, maybe thousands, of specific PCR primer pairs, each pair designed to amplify one small segment of one of your target genes. You throw all these primers into one reaction tube with the patient's DNA and run the PCR. It simultaneously makes millions of copies of just those targeted regions.
That sounds faster, maybe?
Generally, yes. Multiplex PCR is often faster and can be more efficient for smaller, well-defined gene panels where speed is really critical, like getting results back quickly for a cancer patient needing treatment decisions.
So labs choose between capture and multiplex PCR based on the panel size, the speed needed, cost, etc.
Right, but the end goal of both is the same. Focus the sequencing power only on the genes you care about for that specific clinical question.
And the big advantage of doing that, of using a targeted panel.
You maximize your sequencing depth on those crucial genes. Instead of spreading your reads thinly across the whole genome, you concentrate them. This gives you much higher sensitivity to detect variants, especially those low-frequency somatic mutations in cancer. And it keeps costs down and makes the bioinformatics interpretation more manageable compared to whole genome.
Which is key for personalized medicine and oncology, right? Finding those specific mutations that predict response to targeted drugs.
Absolutely fundamental. That's the bread and butter of many cancer panels. You're looking for things like HER2 gene amplification in breast cancer, which predicts response to drugs like trastuzumab, or EGFR mutations in lung cancer, guiding the use of EGFR inhibitors.
Finding the Achilles heel of that specific tumor.
Precisely. And it's not just a one-time thing. The source material highlights that repeat testing is becoming more common.
Why repeat the test?
Well, cancers evolve, especially under the pressure of treatment. A patient might respond well initially, but then the disease comes back. Sequencing the tumor again at relapse can reveal new mutations that confer resistance to the first drug. That knowledge is critical for choosing the next line of therapy. It's dynamic.
Guiding treatment throughout the patient's journey. Okay, so targeted panels are the workhorse. What if you need a broader view?
Then you move up the scale. The next step is usually whole exome sequencing, or WES.
Exome. That's the protein coding parts of the genes.
Yes. WES is still technically a targeted approach. You're using hybrid capture, usually to enrich for just the exons, but it's a much, much bigger target. It covers roughly 1.5% of the entire genome, but that 1.5% contains the vast majority of genes that actually encode proteins, and therefore where most known disease-causing mutations are found.
And where has WES made a big impact?
Hugely impactful, particularly in diagnosing rare genetic disorders. Think of kids with complex, unexplained syndromes often called orphan diseases. For years, they might go through a painful diagnostic odyssey. Countless tests, no answers. WES has been revolutionary in finally providing diagnoses for many of these families, identifying the causative gene mutation, and sometimes even pointing towards potential therapies.
Giving answers where there were none. That's powerful. Okay, panels, exome.
Yeah.
What's the top tier?
The most comprehensive approach is whole genome sequencing, WGS.
Sequencing literally everything, all 3 billion base pairs?
Pretty much. The 1.5% exons plus the other 98.5%, the introns, the vast stretches of DNA between genes, all of it.
When would you need to do that? If WES covers most disease mutations?
Currently, WGS is mostly reserved for specific situations. Often, it's used when WES has already been done and came back negative, but there's still a very strong clinical suspicion of a genetic cause. Maybe the mutation lies in a regulatory region outside the exon, or involves a type of change WES isn't good at finding.
What kind of changes can WGS find that WES might miss?
The big one, as we touched on earlier, is its unique capability among current NGS methods to reliably detect certain types of novel structural rearrangements.
Those large insertions, deletions, translocations. Because WGS gives you more context across the whole genome.
Exactly. Because you're sequencing everything, you have a better chance of finding reads that span the breakpoints of these large structural changes, allowing the bioinformatics tools to piece together what happened. Short-read WES can struggle with that if the breakpoints fall outside the targeted exons.
But WGS isn't the standard go-to test yet.
No, mainly due to practical hurdles. The cost is still higher. The sheer volume of data makes the bioinformatics analysis much more complex and time-consuming, especially interpreting variants in that vast non-coding space where we often don't know the function.
So turnaround times are slower.
Right. And that interpretation challenge is significant. So while incredibly powerful, WGS isn't yet the routine workhorse for most clinical questions. It's more of a specialized tool for complex cases or research. You know, what's really fascinating, and the source highlights this, is the theoretical potential here, because NGS platforms fundamentally could detect almost any kind of genetic change, from the tiniest single-base pair swap in SNT all the way up to huge chromosomal abnormalities like gaining or losing entire chromosomes, aneuploidy.
So the underlying tech has the potential to do it all.
In theory, yes, which strongly suggests that as the technology continues to improve, costs drop, read lengths get longer, accuracy gets even better, NGS could eventually replace almost all the different genetic testing methods we currently use. Karyotyping, FIH microarrays, single-gene tests, they might all converge onto NGS platforms.
A unifying technology for genetic diagnostics.
That seems to be the direction, and as it matures, we're seeing NGS being applied in exciting new ways, beyond just sequencing a patient's genomic DNA.
Like what? What are the new frontiers?
Well, analyzing RNA is a big one. We call it RNA-seq, or transcriptome analysis.
Sequencing RNA instead of DNA. Is the process very different?
The basic NGS chemistry is quite similar. You just need an extra step at the beginning to convert the RNA into more stable complementary DNA, or cDNA, first. But after that, sequencing cDNA is straightforward.
And why would you want to sequence RNA instead of DNA? What information does it give you?
RNA analysis tells you about gene expression, which genes are turned on or off in a cell, and at what level. That's critical for understanding cell function and disease states. Plus, sometimes RNA analysis is just practically better for detecting certain things, especially fusion genes resulting from chromosomal translocations.
Ah, like the example given in the source, the BCR-ABL fusion in chronic myeloid leukemia, CML. Why is RNA better there?
It comes down to the structure of the genes involved. The actual DNA breakpoints for the BCR-ABL translocation often occur within very large introns, those non-coding sections within the genes.
Introns can be huge, right? Thousands, even hundreds of thousands of base pairs long.
Exactly. So our gut microbiome, skin microbiome, it's incredibly complex. Trying to culture all those organisms was impossible. But with NGS, you can just take a sample, extract all the DNA present, human and microbial, and sequence everything. Then bioinformatics tools sort out which sequences belonged to which microbe. It's opened up a whole new understanding of how these microbial communities affect our health and disease.
Fascinating. And you also mentioned something earlier that sounds very futuristic. Circulating free DNA, the liquid biopsy.
Right, cfDNA. This is arguably one of the most exciting clinical applications right now.
So this is finding tiny bits of DNA floating in the bloodstream. Where does it come from?
Mostly from cells that are dying through a process called apoptosis. They release fragments of their DNA into circulation. In healthy individuals, there's a low level of cfDNA. But in patients with cancer, for example, tumor cells are also dying and shedding their DNA, including DNA carrying the tumor-specific mutations into the blood.
So you can potentially detect cancer mutations just from a blood draw without needing a tissue biopsy.
That's the principle. It's non-invasive or minimally invasive. And it's not just for initial detection, although that's an area of intense research for screening. Where it's already transforming care is in monitoring treatment response and detecting resistance.
How so?
Imagine a patient starts therapy. You can take blood samples periodically and use highly sensitive NGS to measure the amount of that tumor-specific mutant DNA in the cfDNA. If the therapy is working, the level of tumor cfDNA should plummet.
Oh, okay.
If the levels start to rise again, it might mean the cancer is starting to grow back, potentially even before it's visible on imaging scans.
An early warning system.
Exactly. And even more powerfully, you can sequence that cfDNA to see if new mutations have emerged in the tumor, mutations that might confer resistance to the current therapy.
Giving you real-time information to potentially switch treatments much earlier.
Precisely. It offers a dynamic, real-time window into the tumor's evolution without needing repeated invasive biopsies. It requires incredibly sensitive NGS because the amount of tumor DNA in blood can be tiny, but the technology is getting there.
That sounds incredibly powerful. Are there even newer sequencing technologies on the horizon beyond current NGS?
Yes. What people are starting to call third-generation sequencing, sometimes also kind of single-molecule sequencing, or maybe confusingly, next-generation sequencing.
Okay, if second-gen NGS brought massive parallelism in short reads, what's the big leap for third-gen?
The defining feature is that these new technologies aim to sequence single individual molecules of DNA or RNA directly in real time without needing that local amplification step, the bridge PCR we talked about.
Skipping the cluster generation, why is that significant?
Several potential advantages. First, it simplifies the sample preparation quite a bit, which could make things faster and cheaper. Second, PCR amplification, even localized, can sometimes introduce biases. Some sequences might amplify more efficiently than others. Sequencing single molecules directly avoids that potential bias. And third, some of these third-gen technologies promise much, much longer read lengths than standard NGS. We're talking tens of thousands, even hundreds of thousands of base pairs in a single read.
Wow, super long reads. That would overcome the problems short reads have with repetitive regions and large structural variants, wouldn't it?
That's the hope, yes. Longer reads make genome assembly and structural variant detection much easier. The accuracy of some early third-gen platforms was a bit lower than standard NGS, but it's improving rapidly.
So these technologies are starting to move into the clinical space.
They are definitely knocking on the door. They're being used more in research now, but expect to see third-generation sequencing making a significant impact in clinical labs relatively soon. It could further accelerate diagnostics.
Okay, let's try to synthesize this. It's clear that NGS platforms have just completely changed the game in diagnostics. We went from looking at maybe one gene, one target at a time, usually needing a pretty clean sample, to being able to analyze millions, even billions of different DNA or RNA molecules simultaneously, even from complex, messy samples, all while costs have plummeted.
It's a paradigm shift, no question. Complex molecular data is now routine. It's right at the heart of clinical decision-making in many areas.
But the crucial takeaway, I think, is that NGS alone isn't enough. It generates the data, yes, but it's the partnership with bioinformatics that unlocks the meaning.
Absolutely inseparable. Bioinformatics provides the algorithms, the pipelines, the databases, the interpretation framework, everything needed to translate those billions of raw sequence reads into actionable clinical insights about disease risk, diagnosis, prognosis, and treatment selection. Honestly, at this point, the bottleneck often isn't generating the sequence data anymore. It's the interpretation, making sense of it all.
And we also touched on the fact that despite this incredible power, NGS isn't perfect yet. There are still certain types of genetic lesions.
Right, those really large repeat expansions like in Fragile X or maybe some very complex rearrangements or even just kilobase scale deletions. Sometimes older, more established methods like southern blotting or specialized hybridization techniques can still be more reliable or straightforward for those specific cases.
So choosing the right test for the right question. The clinical judgment is still paramount. It's not just run NGS on everything.
Not yet, anyway. Test selection remains nuanced.
Which brings us to a final provocative thought for you, or a listener, to consider.
Given this trajectory we've discussed, NGS getting cheaper, faster, read lengths getting longer, third-gen tech emerging, and the increasing ability of NGS platforms, coupled with advanced bioinformatics, to potentially detect all types of genetic lesions, from SNPs to large structural variants, maybe even epigenetic marks like methylation.
Where does that leave the traditional role of, say, the diagnostic molecular pathologist or the cytogeneticist?
If the platforms eventually handle virtually all the detection, does the human expert role shift almost entirely away from performing the physical assays? Will their primary function become being a high-level bioinformatics interpreter, someone whose main job is to critically evaluate the vast amounts of data and the outputs of complex algorithms, perhaps even AI-driven analyses, to ensure the final clinical interpretation is accurate and sound?
And if that is the future, what specific steps, what kind of training or oversight would you think is necessary? How do we ensure that essential human critical thinking, that clinical correlation, remains absolutely central to the diagnostic process, especially when we're dealing with conclusions drawn from billions of data points potentially analyzed by machines?
Something to mull over. How do we leverage this incredible technology while keeping human wisdom firmly in the driver's seat?